this is going to be my review session for Exam 2, Anatomy and Physiology. In this video, we're going over the integumentary system, the skeletal system, the muscular system, and just a little bit of the central and peripheral nervous systems. We've only done like one lecture on that so far, so we're just going to go over sort of just the basic difference between both of those. That's all we need to know. And yeah, that's going to be chapters 5, 6, 7, and 8. So let's get into it. I'm gonna see if I can just skip all the way down to chapter 5. There it is, perfect. All right, chapter 5 is the integumentary system. Now, in that we have the cutaneous membrane and then the accessory structures. The cutaneous membrane is going to have the epidermis and the dermis in it. And then the accessory structures, that's going to be your hair follicles, your exocrine glands, and your nails. Yeah, we got your integument, which is your skin, your cutaneous membrane, and then the derivatives of the skin, which are your nails, hair, sweat glands, and sebaceous glands. Now we need to know some of the functions of the integumentary system. Pause right now and see if you can try and remember any. All right, so we've got protection, prevention of water loss, temperature regulation, metabolic regulation, and that's going to be UV radiation, vitamin C production, calcium, and phosphorus absorption. Another function is going to be immune defense. That is your epidermal dendritic cells. Next is your sensory reception. Those are your Merkel cells. And lastly is excretion and secretion. This is going to be your perspiration and then your sebaceous glands, which produce sebum. All right, now let's go into each one of those and give you a little bit more detail. So for protection, your integumentary system protects your body from physical trauma. You get punched, it doesn't go straight to your muscles. You have to go through your integumentary system first. It protects you from pathogens, so you don't get sick super easily, from external toxins, and from UV rays. The functional, that's tanning. Okay, we've also got to look at prevention of water loss. Most water is prevented from leaving the body thanks to the integumentary system. The exceptions to this, are sweat and urine. Next, temperature regulation. You have vasoconstriction and vasodilation. If you can break those down, constrict, gets smaller, dilation gets bigger. That's going to be your veins. So vasoconstriction, when it gets smaller, it prevents heat loss because your veins are going to stop going to like the very surface of your skin and releasing heat that way. It's going to stick deeper towards your body and keep you a little bit warmer. Vasodilation is the opposite of that, where those veins, they get a little bit bigger and the blood goes through that, releasing heat to the outside world. That's why your skin gets flush on a hot day. You get pale when it's cold and you get a little red and sweaty when you're hot. All right, metabolic regulation. Honestly, I don't really understand this one too much, just a little bit. This is UV radiation, which in turn produces vitamin D, and that, becomes calcium and phosphorus absorption. I'm not going to remember that one. Another function is going to be immune defense. This is your Langerhans cells, your epidermal dendritic cells. Sensory reception, those are your Merkel cells. An example of this is your nerve endings from temperature, touch, texture, vibration, sensation, all of those. If a ladybug lands on you, you can tell. And those are thanks to your Merkel cells. And lastly, exocrine secretion. This is perspiration. Let's see, sebaceous glands. Yep, those produce sebum for lubrication of skin and hair. So those are your functions. There are 7 total. You probably don't need to remember all of them, just remember a few. So let's get into the structure of the integumentary system. Integument is skin. It is the largest organ of your body and makes up 7 to 8% of your total body weight. Very interesting, right? They ask you what the largest organ is, it's going to be your skin. Composed of, the integument, your nails, hair, sweat, and sebaceous glands. There are 3 layers to your integumentary system. Actually, this might be a little wrong because I don't believe your hypodermis is a part of your integument. Yeah, it's not. Okay. So 3 layers to it, though, to your skin. It's going to be your epidermis, your dermis, and your hypodermis. Your epidermis, that is going to be the most superficial layer, closest to the surface. It is the upper region of epithelial tissue. Dermis, that is the deeper layer to that, and it is the thickest layer. It is just underneath your epidermis. And then, lastly, we have your hypodermis. That is the deepest layer. Not necessarily the thickest, just the deepest. It's the lowest. It's also known as your subcutaneous layer. And remember, out of these 3, your hypodermis is not considered part of the integumentary system. So that might be a little test question right there. Epidermis. So cells of the epidermis, you have your keratinocytes, your melanocytes, your dendritic/Langerhans cells, and your tactile Merkel cells. Keratinocytes, they produce keratin, which is a fibrous protein. They make up most cells of the epidermis, and they're tightly connected by desmosomes. Next are your melanocytes. Those make up about 10 to 25% of cells in the deepest layer of your epidermis. They produce pigment called melanin, which get packaged into melanosomes. They protect the apical surface of keratinocyte nucleus from UV damage. Woohoo, okay. So your dendritic cells, macrophages, key activators of immune systems. So remember your dendritic Langerhans cells, they are at you. They protect you from toxins, from pathogens. Yeah, so those are your immune defense cells. You can try and remember it like Langerhans, not today. I'm not going to get sick today. Later, Hans. It'll work. And then lastly, your Merkel cells. Those are your sensory and touch receptors. All right, your epidermis, keratinized stratified squamous epithelium tissue. It is the most superficial, between the epidermis, the dermis, and the hypodermis. And it is avascular. You know what avascular means? There's no veins going through it. And I think it's the only one that is. It is composed of 4 to 5 stratums, layers, which go as follows from deep to superficial. The deepest is going to be your stratum basal. After that is your stratum spinosum. The spinosum is a little bit of a spiny layer. It's got those. I don't know what you call that. It's like little hills and notches. It's a prickly layer. After that, this is your middle layer. Just about. It is the stratum granulosum. After that is your stratum luciderm. Those are only in thick skin, which is the palms and the soles. Palms of your hands, soles of your feet. And then the topmost layer, stratum corneum. It is the dead, horny layer. Not quite sure what that was. 20 to 30 layers. Yeah, every cell in the stratum corneum, it's died, and I'm pretty sure all of its organelles have been recycled, and it's mostly keratin at that point. So let's get a little bit more detailed into this. Your stratum basal, that is the deepest layer of the epidermis. It is a single layer of cuboidal to columnar cells. Is attached to your basement membrane. So the cells that are in there are keratinocyte stem cells. So that's where your keratinocytes are going to be produced, and then subsequently goes up each layer until it gets to the corneum. So your keratinocyte stem cells, they constantly replicate to reduce the keratinocytes found in all other strata, and they produce keratin proteins. Next, your stratum spinosum. Superficial to stratum basal, it appears tiny in slides. Cells, there are several layers of keratinocytes attached together by proteins. And Langerhans cells, which fight infections and cancer cells. Stratum granulosum. This is superficial to the stratum spinosum. It has 3 to 5 layers of keratinocytes, and keratinization begins here. Stratum lucidarum, superficial to stratum granulosum and only found in thick skin. Stratum corneum, this is the most superficial layer. It's the visible layer. You look at yourself, you're like, oh, what is this? That's your corneum. And it consists of 15 to 30 layers of dead, scaly, interlocking keratinized cells. Thicker in thick skin than it is in thin skin. You're going to see in cross sections that it's going to be really thick in your thick skin. It's going to have like a big versus a very thin line of it. And then, when you're exfoliating, that's what you're getting rid of, is a lot of your dead cells from the corneum layer. I feel like a good mnemonic for this is cute ladies get special blankets. Corneum, lucidarum, granulosum, spinosum, basal. All right, so keratinization. That is the process by which cells are filled with keratin. Your cell's nucleus and organelles disintegrate and cells begin to die. What remains is keratin interlocked by a phospholipid layer, and it is completed at the upper level, the stratum corneum. Thick skin, location, palms, fingers, toes, and soles. Palms, fingers, toes, and soles, toes, and soles. It has 5 layers, as opposed to thin skin, which only has 4. Thick skin, it contains lucidum, sweat glands only. That means no hair follicles or sebaceous glands. Yeah. So you can sweat from your palms, but you're not going to have hair coming out from there, or sebum coming out from there. Same with the bottoms of your feet. If you do have hair or sebum, that's a little interesting. That's cool. Location for thin skin, that is going to be the rest of your body. Everywhere else, you're going to have 4 layers, lacking the lucidum layer. These, the thin skin, it contains hair follicles, sebaceous glands, and sweat glands. Epidermal variation. Skin color is determined by 3 pigments. Hemoglobin, that determines whether you're a little bit more pink or blue. Melanin, the number of melanocytes is the same, but the production differs. So UV light encourages melanocyte activity to produce melanin. And then keratin, that is the yellow-orange pigment from food, and that builds up in your stratum corneum. Skin markings. You have nevus, hemangiomas, and friction ridges. Nevus, that is an overgrowth of melanocytes, aka moles. Unlike freckles, which are from increased melanin production, this is from increased melanocytes themselves. Hemangioma, that is the proliferation of benign tumors. That's going to make strawberry marks, and they may disappear in childhood. Friction ridges, those are folds of epidermis for grasping. So that's going to be your fingertips. We need some friction to be able to hold things and, grip. So those are folds of your epidermis, specifically for grasping. Epidermis accessory organs. Structures that grow from invaginations of the epidermis into the dermis. I'm not sure if that's a real word. Invaginations. I might have made that up. Those are going to be your nails and your hairs. So, nails. Those are derived from the stratum corneum again. Cells densely packed with parallel fibers of hard keratin. So remember, nails, those are parallel fibers of hard keratin. Sometimes if you look down and you've had some trauma done to your fingernail in like a certain spot where the nail is produced, you'll see that when it grows out, there's going to be a ridge or something that continues in that same spot. You can think of that for the parallel fibers because it's always going to be going in that same direction. Hairs. Those are columns of keratinocytes growing from follicles. Those are produced in the dermis or the hypodermis, and there are 3 regions of a hair from deep to surface. That's going to be your hair bulb, your hair root, and then your hair shaft. Shaft is going to be the little part that sticks out of your skin. The root is inside, and the bulb is surrounding that. Hair nails are both composed of dead keratinized cells growing from an area of replicating cells that are compressed together. Nails grow from the nail matrix, and hair grows from the hair matrix within the hair follicle. The main differences are going to be the rates and amount of keratinization. Functions of hair. So you're going to have 5 different functions here: protection, heat retention, sensory reception, visual identification, and chemical signal dispersal. So an example of protection is going to be your ears, your eyes, and your nose. Your eyes, you have eyelashes. The whole reason we have those is to prevent stuff from getting in there and making us go blind. Your ears, same there. I mean, it's probably stop stuff from getting in, but also might help diffuse really loud noises, potentially. I'm making that up, potentially. And then same with your nose. You have hairs there that are helpful in stopping not just debris, but also pathogens and things like that from getting up there. Heat retention. Yep, that's your hair coming out of your head. Helps keep in the heat there. Sensory reception, light and touch. Sensory reception, light, touch. Oh, yeah, so like on your arm, if you barely hover over it and you touch just the hairs, you can feel that. They don't have to touch your skin, they can touch your hairs, and you'll know that something is there. It's like a whisper in the night. Shh. Visual identification. Yeah, your hair can indicate your age, your sex, your gender. Help people identify their friends and whoever they're talking to. Otherwise, if everybody's bald, they're going to look pretty similar. Chemical signal dispersal. So your hair also helps disperse your pheromones in axillary and pubic regions. So some more notes about your hair. Hair is a thread-like structure consisting of columns of dead epithelial cells filled with keratin. Hair shaft that projects from the skin, from your corneum. And then you also have your hair root and the hair bulb, which are the remaining portions below the skin. Hair follicles. So that structure surrounds the hair shaft, composed of epidermal cells. Infolding of the epidermis into deeper layers, and it is a reservoir of epidermal cells. The hair follicle also contains the hair matrix from which the strand of hair grows. The erector pili muscle. Okay, so this attaches the dermal papillae to the hair follicle. It anchors the hair follicle, and it contracts to pull the follicle upright, causing goosebumps. So your erector pili is what causes your hairs to stand on end. Hair color, caused by production of melanin in the hair matrix. Color variation is caused by different shades of melanin and different rates of production of that as well. Gray hair, that is gradual reduction of melanin production, and white hair is no pigment at all produced. All right, skin exocrine glands summary. This contains your sebaceous glands and your sweat glands. Sebaceous glands produce oily secretions at the hair, and then sweat glands contain myoepithelial cells, which contract upon nervous system stimulation into ducts. So, merocrine, ecrine, abundant on palms, soles, forehead, and function for thermoregulation. They are regulated by the sympathetic nervous system, your fight or flight response. Next, your apocrine glands. They release onto your hair. It is the ceruminous glands, which produce earwax, and then your mammary glands, which produce milk. So, sweat glands, those are distributed all over the skin except for your nipples and part of the external genitalia. Cells of the sweat glands contract in response to nervous system stimulation to push sweat into ducts. There are two types of sweat glands, your eccrine and your apocrine. Your eccrine, also known as merocrine, sweat glands release secretions directly onto the surface of the skin through sweat pores, mostly water with some salt. The highest number are in your palms, sole, and forehead, and it's used to maintain your body temperature. So remember, your eccrine releases sweat onto your skin. Apocrine. Okay, so your apocrine glands, these release secretions directly onto your hair follicle. These are thick organic secretions, which are proteins or lipids, and they react with bacteria on the skin to cause body odor. These are located in your armpits, genitalia, and external ear, and those become active after puberty. So your apocrine sweat glands, those are going to be the sweat glands that release onto your hair, as compared to eccrine, which is onto your skin, but they both release sweat. Your modified sweat glands, those are going to be your ceruminous, which secrete earwax into the ear canal and lubricate the eardrum and ear canal. And then your mammary glands, which in the breast produce milk, and it's only in pregnant and lactating females. Next, your sebaceous glands. Those release sebum, usually directly onto your hair follicle. If you remember, sebum is like a waxy, oily secretion, and this acts to keep your skin and hair moist, and they're activated during puberty. Blocked glands can lead to black and whiteheads, and inflamed glands can lead to acne. So a good way to remember this one is sebaceous glands. It's like sebum. Sebaceous sebum sounds pretty similar. All right, we're going to move on to the dermis. So, the dermis is deep to the epidermis. It has two layers of connective tissue. One is the papillary, the next is the reticular. The papillary layer is superficial to areolar tissue, but it is deep to the stratum basal. It has dermal papillae and epidermal ridges. And then surface area also contains Meissner's corpuscles. Next is the reticular layer for connective tissue. This is deeper, dense, irregular tissue. It's the majority of the dermis, and it contains blood vessels, glands, hair follicles, and nerves. In this layer, you're going to have your Pacinian corpuscles. Next, your hypodermis. This is your subcutaneous layer. It is deep to the integument, and it contains areolar and adipose connective tissue. It is also highly vascularized, which means you have a lot of veins running through it. The functions of the hypodermis are it protects underlying structures, it stores energy, and it works for thermal insulation. Other changes to the integument. Okay, so you're going to have stretch marks, which are called striae. When your stretch exceeds capability, collagen tears. Well, I don't know if I really have to explain stretch marks. For myself, I don't. Wrinkles, that is your diminishing of flexibility and thickness. Comes down to DNA, routine movement, and UV exposure. And that's going to do it for the integumentary system. Woohoo! Chapter 6, Bones. All right, intro to the bones. They are actually living organs with all four tissue types. They are primarily osseous tissue, Smooth muscle in blood vessels, fluid connective tissue proper, cartilage, epithelial lining in blood vessels, nervous tissue, nerves, extracellular matrix is sturdy, rigid rather than fluid, and then calcification. All right, so your bone tissue contains osteocytes, fibers, and matrix. There are two types of bone tissue: compact bone, which is going to be on the outside of most of your bones, and then spongy bone, which is deep to that. There are two levels of structure: your gross anatomy and then your microscopic level. Bone tissue, compact bones. These ones are very solid and dense. They're located on external surfaces of bones. Only tiny spaces are present here. Spongy bone, they are very light and porous. They're internal to compact bone, deep to compact bone, and there are many large spaces present. If you think of a sponge and you think of all the holes in it, that's exactly what this is. It's highly vascularized. There are 206 named bones in the skeleton, and your skeleton is divided into two groups: the axial skeleton and the appendicular skeleton. The axial skeleton is the long axis of your body. It's going to be your skull, your vertebral column, and then your rib cage. And then appendicular skeleton is everything else. It's like your appendages, your arms, your legs, whatnot. The girdle attaches limbs to the axial skeleton. Functions of bones. Okay, so know these. There are 5 functions of the bones: support, protection, mineral storage, blood cell production, and movement. Bones support the body's infrastructure. They protect the surrounding organs. They store minerals, i.e., 98% of the body's calcium is stored in bones. And they produce blood cells. The bone marrow produces new blood cells, hematopoiesis. And then movement. Muscles pull on bones. If you can imagine our bodies without bones and muscles trying to move us, it's not really going to work. You're going to be like a big blob. So support, protection, mineral storage, blood cell production, and movement. Bone classification. They're classified by... Oh, my bad. I was thinking naming. All right, so bones are classified by shape. You're going to have your long bones, short bones, flat bones, odd bones, and irregular bones. Your long bones, they're longer than they are wide. They're like limb, foot, and hand bones. So you got your femur, you got your humerus. Those are all going to be long bones. Short bones, often they're cube-shaped. They vary in size and number. And an example of these are going to be your wrist and ankle bones, your tarsals and your carpals. You know how those look. That's an example of that. Flat bones, they are thin, flat, and slightly curved. Or, slightly curved. Example of this is going to be your sternum, your scapulae, your ribs, and most of your skull bones. Next, odd bones. Varied in size and don't fall into any groups. There are 2 types: sesamoid and sutural. Lastly, we have irregular bones. They are complicated shapes, such as your vertebrae and your coxal bones. Anatomy of the typical long bone. So there's going to be 3 parts to it that you're going to want to remember: your epiphysis, your diaphysis, and your metaphysis. Your epiphysis is the rounded end of the bone. Epi, just remember that a little bit. Rounded end of the bone. Diaphysis is the long cylindrical shaft. It contains the hollow medullary cavity, and it's filled with yellow bone marrow. Metaphysis, area between the diaphysis and the epiphysis. It contains the epiphyseal line, which is hyaline cartilage plate remnant from childhood bone growth. When you're a child, a lot of your bones, they're not fully solidified together. This allows for growth, and once you have finished growing, the epiphyseal line is where that hardens. And tissues of typical long bone. So you're going to have compact bone that is along the external surface, spongy bone, which is within the epiphysis and lining edge of the medullary cavity, and hyaline cartilage, which covers articular surfaces, aka surfaces on the epiphysis that articulate with other bones. There are coverings lining the external and internal surfaces of bone. Bone coverings. The periosteum lines the outer surfaces of bone. Attached to bone by collagen. Remember, peri is basically like around, surround. So that's a good way to remember that the periosteum is what lines the outer surfaces of the bone. So the periosteum also contains osteoblasts, which secrete matrix for bone formation, and they contain fibroblasts for collagen attachment fibers. Contains many vessels and nerves. Cells of bone tissue. Osteocytes. This is the main blood bone cell. They are the mature bone cells, and they secrete bone matrix. Osteoblasts, these build bone. They secrete bone matrix as well, and eventually become osteocytes. Osteoclasts, these destroy bone. They break it down, and they allow for bone remodeling and demolition. There are two large multinuclei, and it helps with the resorption of nutrients, releasing calcium plus phosphorus to the body. So osteocytes, those are the main bone cells. Osteoblasts, those build bone, and then osteoclasts, they destroy it. Bone coverings, the endosteum and endosteum. The endosteum lines internal bone surfaces. It lines the medullary cavity of long bones. It lines the large porous spaces of spongy bone, the trabeculae, and it lines canals through compact bone. It is a thin vascular membrane of connective tissue and contains osteoblasts and osteocytes. That is the endosteum. Next, we have the microscopic organization of bones. We have osteons. This is the basic structural and functional unit of mature compact bone. Just remember the Haversian system for that. And cylindrical structures, they're cylindrical. They form cylindrical structures that run parallel to the shaft of the bone. It's formed around a central canal that houses blood vessels, which provide blood and nutrients, and it's all living tissue. Osteocytes. Osteons are produced by cells called osteocytes. Osteoblasts in the endosteum and periosteum migrate into bone and become trapped in the matrix they secrete, in a lacuna. Osteocytes create rings of bone matrix around the central canal called lamellae. And then osteon components, lamellae, we're going to get into that now. The concentric lamellae, rings of bone tissue around a central canal. We have interstitial lamellae. It is incomplete lamellae, not a part of the complete osteon. It fills gaps between the osteons and remnants of osteons cut by bone remodeling. Circumferential lamellae, just deep to the periosteum and superficial to the endosteum. They extend around the entire surface of the diaphysis and resist twisting of long bone. And then osteon components, canals. We have the central canal, Haversian. These are your blood vessels and nerves. They form the center of the osteon. We have your perforating canal. These are perpendicular connections to the central canal. These transmit blood vessels from the periosteum to the central canal. And then caneculae. These connect bone lacunae to each other and to the central canal. This distributes materials to all osteocytes. All right, so in the picture here we have some bones. On the left we have compact bone, and on the right we have spongy bone. You can tell because it's very porous, all holes in it. Each one of these long rings, that's going to be an osteon. So we have one right here, And then another few over here. I wish you could see my mouse, but you can't. But yeah, the big O shapes, that entire thing is an osteon. Inside of each of those, you're going to have a central canal, which houses nerves, arteries, and veins. So your lamellae, those form the rings in each of your osteons and around all of them in your compact bone. So you have your circumferential lamellae, which go around the entire thing, and then your concentric lamellae, which are inside each of the osteons, as well as your interstitial lamellae, which are between the osteons. Each lamellae contain multiple lacunae. If you look at the image, they're going to be the red bumps, which are along each of the rings of the lamellae. And then if you look at the lacunae, those contain your osteocytes. Another little thing to look at here is, let me get to it so I can zoom in. If you look closely, you can see your canaculae, which are the little lines between your lamellae and your lacunae. While we're looking at this, another good thing to remember is that your central canal that runs vertically parallel to your osteons, and then your perforating canal, those are going to be the canals that are horizontal, running perpendicular to your osteons. So central canals versus perforating canals. All right, back to the notes. Remember, you have your central canals, which contain blood vessels and nerves, and those form the center of the osteons. And then your perforating canals. Those are perpendicular connections to the central canals, and they transmit blood vessels from the periosteum to the central canals. And then your canaliculi, those connect the bones lacunae to each other and to the central canal, and they distribute materials to all the osteocytes. Now, microscopic organization of bone. We're going to get into spongy bone. Though the spongy bone is composed of tuberculi, they are honeycomb-shaped flat pieces of bone and then thin bone plates. They're aligned along points of stress to help resist it, and they contain capillaries in the endosteum, which supply nutrients, lots of open space for red bone marrow, which is the location of hematopoiesis. This is going to be the tuberculi right here on the right. The tuberculi does not contain osteons, and it's irregularly arranged lamellae and osteocytes, which are interconnected by canaliculi, or canaculi. Can't say that. Bone matrix. It's 1/3 organic and 2/3 inorganic. So the organic part, that is your osteoid tissue. It has cells, collagen fibers, and ground substance, and it is resistant to stretch. Inorganic bone salt crystals, which are calcium phosphate, those are resistant to compression, so they're very hard. Bone growth. Interstitial growth, which is growth in length that occurs at the epiphyseal plate. Calcification at adulthood to become the epiphyseal line. Appositional growth is growth in diameter. So we've got interstitial growth, which is length. Remember, I, it's very long. And then appositional growth, A, it's a little bit wider than an I. So it's growth in diameter. Osteoblasts in the periosteum form external circumferential lamellae, and osteoclasts in the endosteum remove bone from the medullary cavity something. All right, next we're going to talk about bone remodeling. Bone continues to grow and renew throughout your life. 20% of the skeleton is replaced yearly. Isn't that insane? Every year, 20%. So, childhood and puberty, it's the formation of new bone, which is way faster than resorption. So, childhood, puberty, you're growing a lot, you're going to be forming a new bone all the time at, a very high pace. And it's going to be a lot faster than the removal of old bone. The first bones to finish growing are going to be your feet. So you'll see like a lot of teenagers with awkwardly large feet. I remember my brother and I, we were in track, and man, we were very awkward back then. Yeah, because feet. All right, last bones are going to be your wrists, and I believe those finish growing. Oh, let me double check actually. Okay, yeah, those finish growing when you're around 16 to 20. All right, so young adults. The formation rates and resorption rates are equal as compared to older adults, where the resorption rates are faster than the formation rates. This is, how you see people start to shrink when they get older. Grandma used to be as tall as you. Now she's a few inches less. So that's why that happens. A note here is going to be osteoporosis, and another note is harder activities like running and pounding of our feet on the ground encourages the body to build stronger and denser bones. So the more you run, the more you do high impact sports, your body is going to be building denser, stronger bones to, work with that. Fracture repair. Spongy bone develops first and then compact bone. It takes about 4 months to a year. Vitamin D, which comes from the sun, and I think you produce it yourself as well. Lack of such can develop rickets, which are soft bones. Rickets are soft bones caused from a lack of vitamin D. Vitamin C, a lack of such can result in scurvy, and that in turn means a decrease of osteoblast activity, which results in weak bones. All right, now that we're done with that, we're going to get into the skeleton. So the axial skeleton is the framework that supports and protects organs in the dorsal and ventral cavities. It contains special sense organs for taste, smell, hearing, balance, vision. Yeah, your big senses. I blanked on that word. And it serves as an attachment site for muscles. So we have this nice image here. We have your skull, all the different bones there. You got your frontal bone, which is at the very front, your occipital bone, very back of your skull, parietal bone. You've got 2 of them on either side. It connects the frontal and the occipital. And then right above your ear, that's going to be your temporal bone. Your sphenoid bone is the bone that touches all other bones in your skull. It's the only bone that does that. Comparatively, you've got your hyoid bone, which doesn't touch any other bone, just cartilage. And then you also have your ethmoid bone. That's going to be superior and deep to your vomer. Let's see, your zygomatic, that is your cheekbones. Your nasal bones will be right here. Your we talked about that. Your maxilla is going to be your upper jaw. So your top teeth, that's your maxilla. And then your bottom tooth, your actual jaw, is going to be your mandible. So we went over all the different bones. Let's see, the cranial cavity is a fluid-filled chamber that supports and protects the brain. So yeah, your brain's sort of floating around there. You've got 2 parietal and 2 temporal bones. And an important one to note is your sphenoid, which articulates, touches all other cranial bones. And then another note, all of these bones, they're going to be attached by sutures. Cranial fossa. The cranium has 3 cranial fossa, distinct depressions that hold the lobes of the brain. Anterior, middle, and posterior. So your anterior cranial fossa is formed by the frontal bones and the sphenoid bone. It holds the frontal lobes of the brain. Your middle cranial fossa, that is formed by the sphenoid and the temporal. So your anterior is with the frontal, and the middle is with your temporal, right there. And that holds the temporal lobes of the brain. And lastly, your posterior cranial fossa is formed by the temporal and the occipital bones, and that holds the brainstem and the cerebellum. So frontal, temporal, and then brainstem and cerebellum. Facial bones. The skull contains 14 total facial bones. The paired bones, and then there's single bones. So paired, that's going to be your maxillae, your palatine. Your palatine is going to be the hard upper palate of your mouth. Nasal, that's your nose bridge. You've sort of got it cut in half right there. There's two of them. Your zygomatic cheekbones. Lacrimal, that is part of the eye socket. Your inferior nasal concha, that is your deep nose bone. Don't go sticking your finger up there. And then you have single bones too. That's just going to be your vomer, which is the lower half of your nasal septum. I believe you could be close to seeing that if you look really hard. Cut in. And then your mandible, which is your lower jaw. All right, and then your auditory ossicles. So these are going to be your inner ear bones. So if you look at a diagram of where these are located, it's going to be located in your middle ear, which a lot of people, that's where they get their ear infections at. And that's why it makes it really weird and hard to hear, because it's all inflamed and yeah, it messes up how these function. So your auditory ossicles, in the temporal bone are 3 inner ear bones. Okay, I don't know if I read that. That transfer sound from the external ear to the inner ear. I don't think these are located in the temporal bone, so ignore that. But anyways, there's 3 bones that make up your ossicles. That's going to be your malleus, your incus, and your stapes. Malleus, incus, stapes. Your malleus is going to be the hammer. That's going to be the very first bone, the closest to the outside. That's your malleus. It sort of looks like a hammer. It's straight, and then it's got like a bigger thing on the end. And then you've got your anvil, which is your incus. That sort of looks like an anvil, but I don't quite see it. And lastly is your stapes, which looks like a stirrup. So, if you ride a horse, it's got the U-shaped thing that you put your feet into. That's what that is. And you really just need to remember that your hammer, that's the closest to the outside. And then your stirrup that touches your cochlea, that is the most deep one, and the anvil just connects them. So just remember malleus, incus, stapes. Next, your vertebral column. Now, your vertebral column has 26 bones. You have 24 vertebral. You have 1 sacrum and 1 coccyx. Now, I'm not quite sure about that one because there's cervical, you've got 7, 12 thoracic, 5 lumbar. That's 22, and then 5 sacrum and 4 coccyx. So I guess for my test, it's 26, but that might not be the same case for everybody. There are several functions to this. The vertebral column encloses and protects the spinal cord. It supports the skull. It supports the weight of the head, neck, and trunk. It helps transfer weight onto the lower limbs and the lower body, helps maintain upright posture, and each vertebrae is separated by intervertebral discs, which are mostly collagen? No. Yeah, cartilage. They're composed of cartilage. So, divided into regions from superior to inferior, you've got your cervical at the top, and then your thoracic, that's going to be your upper back, and then your lumbar, that's going to be your lower back. Good way to remember this is cervical, 7. You eat at 7 a.m. Thoracic, 12. You have lunch at noon. And then lumbar, 5. You eat dinner at 5. So 7 o'clock, breakfast, 12 o'clock, lunch, and 5 o'clock, dinner. Next, you have your immovable parts to this, and that's going to be your sacrum and your coccyx. Your sacrum is composed of 5 fused vertebrae, and then your coccyx, 3 to 5. Generally, you can just remember 4. And then next you have your atlas bone. That's going to be C1. That's going to be the very, very, very first one at the very top. The atlas is your first vertebrae. It allows you to shake your head yes. Up and down. Okay. Atlas. There's a way to remember this. Yes. There you go. It articulates with the occipital condyles and make sure that you can identify this bone. Next, the axis. That is C2. It is the very next one down. The axis fits with the atlas with no intervertebral disc in between. The den of the axis fits into the space of the atlas and is the axis that allows you to shake your head no. So if you look at the X, no. There you go. Axis, no. Yes. Okay. Anatomy of typical vertebrae. So you've got the body. That's going to be the biggest part at the very bottom. You're going to have your spinous process, which is going to be the central point sticking out. Next, you're going to have your articular process, which is the next one down from your spinous. And the ones that stick sort of this way, horizontally away from each other, that's going to be your transverse process. In the center of all of this, you're going to have a big hole, and that's going to be the vertebral foramen. That's where a lot of that's where like your spinal cord and blood vessels and things run through. So we talked about the body, the transverse process, spinous process at the very top, vertebral foramen. Remember foramen, that's a canal, that's like a hole. Your inferior articular facet. I forget where that is. Let's see if I have it anywhere. I don't. We can figure it out. Your superior articular facet. And then, yeah, this is a good way to remember what each type of vertebrae is so you can identify them. So your thoracic, if you look straight at it, it's going to be shaped like a giraffe. You can look at it and be like, wow, that looks like a giraffe. It's got the little horns. It's got like a thing at the top and things going this way. Yeah. It's also heart-shaped depending on which angle you view it at. Next is the lumbar. Remember, that's the most lowest. And that one's going to look like a moose when you look at it. So it's going to have sort of things going out in all directions, not just like the little, oh, that's a cute little giraffe with its horns and ears. No, this has whole antlers to it. Shoo. And then your cervical, you can just tell that because it is so teeny weeny compared to the other ones. So your cervical vertebrae, those are the only vertebrae with transverse foramina. It's the only vertebrae with small holes. And those allow the passage of the vertebral artery, vein, and sympathetic nerves. It supplies blood to the vein. Thoracic vertebrae. These articulate with the ribs. Lumbar vertebrae, they're very thick and oval shaped. Blunt spinous process points directly posterior. So lumbar, go straight back. Sacrum and coccyx. The sacrum is a triangular bone formed by the fusion of 5. Coccyx is a very small tip of the sacrum, and it's formed by 3 to 5 bones. Your thoracic cage, your rib cage, has 2 functions. And for some reason, I only have one put here. Attachment site for muscles, and then I believe the other one should be protection. So true ribs, that is going to be the 1st through the 7th, 1 through 7. Connected directly to the sternum by costal cartilage. The costal cartilage is going to be like right here. It connects it to the sternum. Yeah, and then outside of that. Your false ribs, that's going to be 8 through 12. Those are connected indirectly to the sternum by the first ribs. And then lastly, you have your floating ribs. This is going to be 11 and 12. 11 and 12 are also considered false ribs. Floating ribs are not at all connected to the sternum. So the thoracic cage and the anatomy of a typical rib. So you're going to have let's see, okay. you're going to have the head on one side and then the articulation with costal cartilage on the other side. So the head connects to your spine, and then, yeah, vice versa. You've got your head, neck, tubercle is a little thing that sticks out, and then the articular facet, that is for the transverse process corresponding vertebra. So that's where it connects. Ribs have two main landmarks which are important. The head articulates with the body of the thoracic vertebrae. So yeah, head articulates with the vertebrae. And then the tubercle articulates with the transverse process of the thoracic vertebrae. Your appendicular skeleton. All right, sweet. This includes your pectoral girdle, which is your shoulder girdle. That contains your clavicle and scapula. Your upper arm, which is your humerus, radius on your pinky end. Sorry, ulna is on your pinky end. Radius on your thumb end. Carpals, metacarpals, and phalanges. Next is your pelvic girdle. That contains your coxal bone, your ilium, ischium, and pubis. And then know the difference between the pelvic girdle and your pelvis. Lower limb. That's going to be your femur, patella, tibia, fibula, tarsals, metatarsals, and phalanges. The forearm. All right, so yeah, you've got your ulna that is on your pinky side, and it is the medial bone of the forearm. The olecranon is the elbow. Radius, lateral bone of the forearm. So remember, anatomical position, your thumbs are going to be facing out, so that makes it lateral. Both bones articulate with the humerus and the carpals. Wrist and hand. The carpal bones are the eight bones of the wrist. Remember, there are 8 carpal bones. Fingers are named thumb to pinky. 1, 2, 3, 4, 5 in Roman numerals. The very first finger, your thumb, is going to be called your pollex. So remember that. There are 8 carpal bones, and your thumb is named the pollex. The 5 metacarpal bones articulate with the distal carpal bones and make up the palm of the hand. These are also named with Roman numerals, 1 through 5. And then there are 14 phalanges. 5 for your pinky through your pointer finger, and then I believe, 3 for your pinky through your pointer finger, and then I believe just 2 for, yeah, 2 for your pollex, your thumb. All right, your pelvis, the coxal bones, and the sacrum. So remember, the pelvis is just the coxal bones and the sacrum. The girdle contains pretty much everything else. Nope, I might have had that backwards. The pelvis is the entire bony structure of the lower trunk. It includes the pelvic girdle and the sacrum and the coccyx, while the pelvic girdle specifically refers to the 2 hip bones that form the anterior and lateral parts of the pelvis. So pelvis, that's your whole thing. Pelvic girdle, that's just going to be the 2 hip bones. All right, how to tell apart the male and female pelvis. So the pelvic angle is 1. Female pelvises have a much wider angle, 100 degrees or more, while males have 90 degrees or less. The ischial spine in males is closer, while it's further away in females. The acetabulum faces more inward in males, outward in females, and the tailbone in males points inwards and points outwards in females. So to recap, tailbone, inward, so for male, inward tailbone, inward acetabulum, 90 degree or less pelvic angle, and close ischial spine. Females, outward facing tailbone, acetabulum faces more outward, pelvic angle is 100 or more, and ischial spine is further away. Okay, moving on to the ankles. So the ankles have 7 tarsal bones, which make them up. And the 2 main tarsal bones are going to be your talus and your calcaneus. So your calcaneus, that's going to make up the very bottom of your foot. You can feel it right there. That's what you step on. And then your talus is going to be right above that. That's what articulates with sort of the wrist of the foot. What do you call that? Whatever joint is right there. So calcaneus is on the bottom. That's what you step on. You can remember it by the C. The shape of that sort of looks like the shape of your heel. Calcaneus. So the talus, that articulates with your tibia, and then the calcaneus, that articulates with the heel. So the foot. Toes are named from big toe to pinky, 1 through 5 in Roman numerals. And then your big toe, that's going to be the hallux. So you have 5 metatarsal bones that articulate with the distal tarsal bones and make up the arches of the foot. 5, 1 for each toe. Again, Roman numerals to name them. So you have 14 phalanges, 3 for your pinky through your pointer toe, and 2 for your hallux, your big toe. Now you have classifications of joints, of different articulations. So the 3 big classifications are going to be synarthrosis, amphiarthrosis, and diarthrosis. Synarthrosis, those are going to be fibrous, gomphosis, cartilaginous, bony, fusion, fibrous. I'm not going to go over these because there's so much, and I don't really think it's going to be that important to the test. But if you want to pause and read this, then go ahead. And then, yeah, diarthrosis, those are synovial. Classifications of synovial joints. These ones are just a little bit more important. So you have your gliding joint, hinge, pivot, ellipsoid, saddle, and ball and socket. These ones you can physically understand a little bit more. So the gliding joint, these are multi-directional movement in a single plane. So, a lot of directions you move it single plane though. So hinge joint. These are going to be like your elbows and your knees. Oh my goodness, it's like a hinge. It can open and close like a door. It has angular movement in a single plane. Pivot joint in your neck. So that's going to be like your atlas and axis, pivot joints, rotational movement around a single axis. Another example is in your wrist, between your radius and ulna. It's just like, my head and my wrist. Those are pivot joints. Ellipsoid joints, that's going to be your wrist joints, and between the phalanges and metacarpals. Between your phalanges and your metacarpals, that has angular movement in 2 planes. It has biaxial movement. Next is saddle joint. Those are your opposable thumbs. That's what makes us a little special as humans and monkeys. We got opposable thumbs. It has angular movement in 2 planes and circumduction. And then lastly, ball and socket joints. That is the shoulder joint between the humerus and the scapula. And another example is going to be your hip joints. Those are angular rotation and circumduction. What you really just got to know with these is going to be your hinge joint and then your ball and socket joint. An example of each. So a hinge joint has to be opening and closing, just like your elbows and your knees. And then ball and socket, that's going to be your shoulders, because you can take your shoulder and you can go in a big old circle. Ball and socket joint. Like a shoe. Just like that. All right. And that leads us into muscles. Perfect. Chapter 7, muscles. Characteristics of muscle tissue. There are 4. Excitability, contractibility, extensibility, and elasticity. Excitability is the ability to receive and respond to stimuli. Contractibility, the ability to shorten forcibly when stimulated. Extensibility, the ability to be stretched. And elasticity, the ability to recoil to resting length. Muscle tissue, that makes up nearly half of body's mass. It transforms ATP into direct mechanical energy, so chemical energy into exerting force. There are 3 types of muscle tissue: skeletal, cardiac, and smooth. And then myo, mys, and sarco, those are all prefixes for muscle. The first type, skeletal muscle. Organs attached to bones and skin. So skeletal muscle is composed of elongated cells called muscle fibers. It is striated and it has voluntary contraction, which requires nervous system stimulation. It contracts rapidly and gets tired easily, and it has powerful contractions. Functions of skeletal muscle tissue. 5. So it has body movement and control of openings, mouth and bottom. Maintenance of posture, temperature regulation, storage and movement of materials, and then support. All right, and then there are actually 3 types of skeletal muscle fibers. You have slow, intermediate, and fast. Slow fibers are called slow oxidative, aka red fibers. They're small in diameter. Called red because they have an increased blood supply. They have extended contractions, which means they fatigue slower and they contract more slowly. It takes 3 times as long to contract after stimulation, running or swimming a marathon. So intermediate fibers are going to be fast aerobic. They fatigue slower than fast fibers. And then fast fibers, those are fast anaerobic, also known as white fibers. Fast fibers don't use oxygen. They're white because they have less blood flow or capillaries. They are large in diameter, they have fast, powerful contractions, and they fatigue rapidly. The ratio of muscle fibers will determine the speed and sustainability of muscle contraction. Muscle fiber distribution. Skeletal is composed of all 3 types. It has a single motor unit composed of just one type. Slow fibers dominate your back and your calves, and there are no slow fibers in muscles that require swift but brief contractions, i.e., your eyes and hands. No slow fibers there. Wink, wink. Muscle attachment has, tendons. Tendon, dense, regular connective tissue that connects muscle to bone, usually cord-like in appearance, and sometimes forms a flat aponeurosis, which covers your abdominal muscles. It's like a large white flat sheath. Muscles cross a joint and attach to an articulating bone. Contraction will move one of the 2 attached bones. Each muscle has 2 attachment points, origin and insertion. The origin is the bone that doesn't move, and then the insertion is the bone that does. The anatomy of skeletal muscle tissue from gross to microscopic. So, skeletal muscle, fascicle, muscle fiber, myofibril, and then sarcomeres. So, there's 2 different types of myofibrils, thick and thin, and then sarcomeres are the structural, functional unit of muscle tissue, like boxcars, which make a train, and that which gets put on a shipping ship. And aside to the connective tissue sheaths, those support cells and reinforce the entire muscle. You have your epimysium, which is dense irregular connective tissue surrounding the entire muscle. It may blend in with the fascia. Your perimysium, which is fibrous connective tissue surrounding your fascicles, and then your endomysium, which are fine areolar connective tissue surrounding each muscle fiber. All right, and then back to muscle fibers. So it is a long cylindrical cell. It's very small in diameter, but can be up to 30 centimeters long. So very long, very small. It has multiple peripheral nuclei towards the outside, and has the sarcolemma, which is the plasma membrane, the sarcoplasm, which is similar to the cytoplasm, so you can remember it that way. And then there are multiple modified structures, which are your myofibrils, your sarcoplasmic reticulum, and T-tubules. Now we can look at the picture on the right for just a little bit of a breakdown on what the muscle fiber looks like and how it is put together. So you have your filaments, that is the very smallest part of the muscle fiber. Those make up your myofibrils, which are these things right here, and each of those makes up your muscle fiber. And then inside of these, you can sort of see like your sarcomeres and how that works. We'll go over that in a bit. And then nucleus and your sarcomeres create the striations and whatnot. Myofibrils. One muscle fiber contains 100 to 1,000 cylindrical myofibrils in the sarcoplasm. They extend the entire length of the cell, composed of repetitive groupings of overlapping thick and thin myofilaments. Myofilaments slide across each other to shorten the myobril, and that is muscle contraction. Myofilaments are composed of thick and thin filaments. Thin is from actin protein and associated proteins. Now you can remember actin, thin. Thin filaments are made of actin protein. And then thick filaments is myosin protein. So be right there. Now let's take a look at your thin filaments. They're a twisted double strand of actin protein, and those bear active sites for myosin head attachment during contraction. Two regulatory proteins are tropomyosin and troponin. Tropomyosin covers the actin bonding sites, and then troponin holds tropomyosin in place. Thick filaments that are twice as thick as thin filaments, and they're composed of bundled myosin proteins. Each myosin protein has a head and an elongated tail, binding sites for actin of thin filaments, and then binding sites of ATP. Thick and thin myofilaments are alternated to function together. The sarcomere, smallest contractile protein / unit of a muscle fiber. They align along the myofibril like boxcars of a train. They are composed of thick and thin myofilaments made of contractile proteins. Myofibril banding pattern. So orderly arrangement of actin and myosin filaments within the sarcomere. Thin actin myofilaments are anchored to Z discs. They extend across the I band and partway in the A band. Thick myosin filaments connect to the M line. Those connect to the Z line via elastic filaments made of titin, and then they extend the length of the A band. And then know why muscle fibers are striated. It is the overlap of thick and thin filaments. More is darker, less is lighter. The darkest part, the Z disc, but the darkest part is the Z disc, and the lighter part is the thin filaments with the titin. The M line is the line at the center of the sarcomere. We can see that right here. There's the M line. Then you have thick filaments all over here. Your Z disc is on the right and left sides. That is the very darkest part. Then I can see where the A band is. Your thin filaments, that's the lighter parts over here. Perfect. All right, let's look at the mechanism of skeletal muscle contraction. The sliding filament theory. Muscles control due to the interaction, sliding, of thin and thick filaments in each sarcomere. Shorten the H zone as filaments overlap. I bands disappear and A bands move closer. The motor unit. Skeletal contraction is voluntary. It requires conscious control. The motor unit consists of a single motor neuron and the muscle fibers it controls. In the eye, for example, one neuron controls 2 to 4 fibers. And then one neuron controls 2,000. Wait a second, I need to double check this. Okay, yeah, so in larger muscles, like your calf muscle, aka your gastrocnemius, that is where one neuron will control like 2,000 fibers. But in your eye, where it's very specific, it's a much smaller ratio. And then, yeah, it's like the all or none principle. All muscle fibers controlled by one motor unit contract at once. All right, let's look at the process of skeletal muscle contraction. 1. Activation. This is at the neuromuscular junction. Must be nervous system stimulation and must generate action potential in the sarcolemma. 2. Excitation, contraction, coupling. Action potential propagated along sarcolemma into the T tubules. It has the release of calcium 2 plus from terminal cisternae. Intracellular calcium 2 plus levels rise briefly, and then calcium 2 plus binds to troponin regulatory binding sites on our filaments, freeing up myosin binding sites on the actin. 3. Sliding filament model of contraction. Myosin binds to actin for contraction. Tropomyosin calcium 2 plus moves tropomyosin off of actin. Myosin binds to actin. The power stroke happens. And then actin filaments move. Excitation, contraction, coupling, noted. Sodium entering the muscle cell causes depolarization. Depolarization leads to the release of calcium from storage within the cell, and calcium allows muscle to contract. Myosin head position and power stroke. There are 2 positions, a high energy state and a low energy state. The high energy state is bound to ATP, and it's the resting state. And the low energy state, ATP, is hydrolyzed to ADP and P, and that is during the power stroke. The power stroke, calcium levels increase in cytosol. It binds to troponin, and then calcium troponin complex pulls tropomyosin away from the actin's myosin binding site. Myosin then binds to actin and completes the power stroke, and the actin filament moves. It's pretty good to go watch like a video on how this works. I feel like that would make it a little bit more understandable. All right, the neuromuscular junction. This is the junction between the axon terminal of a motor neuron and the sarcolemma of a muscle fiber. Axon terminal and muscle fiber separated by a gel-filled space, the synaptic cleft. Synaptic vesicles of axon terminal contain the neurotransmitter acetylcholine, ACH, and then the motor end plate is crucial for interaction between the motor neuron and fiber. The motor end plate contains ACH, acetylcholine receptors. And then know this one right here, the activation of the neuromuscular junction. So 1, nerve impulses reach the end of an axon. Calcium channels open up within the axon, causing ACH vesicles to fuse with the membrane. ACH releases into the synaptic cleft. Then ACH binds to receptors in the sarcolemma, and the binding causes sodium to enter the fiber, which then causes contraction. The destruction of acetylcholine. The ACH bound to ACH receptors is quickly destroyed by the enzyme acetylcholine erase. Acetylcholine esterase. So ACHE. This destruction prevents continued muscle fiber contraction in the absence of additional stimuli. All right, next we have exercise and skeletal muscle. So, atrophy, that is the wasting of tissue, reduction in muscle size, tone, and power, disuse of a muscle, which is, a cast or paralysis, happens then. Hypertrophy is the increased number of myofibrils per fibers increasing in size, the result of exercise. Muscle tone, some of the motor units of muscles contract continuously, producing a resting tension called muscle tone. Resting muscle tone stabilizes the position of bones and joints. All right, rigor mortis. So myosin detachment from thin filaments requires ATP. 3 to 4 hours after death, muscles begin to stiffen with weak rigidity at 12 hours post-mortem. Dying cells take in calcium, which then cross the bridge formation. No ATP is generated to break cross bridges. All right, that's perfect. All right, so that does it for the muscle system. Now we're going to switch over to what we have so far on the central nervous system as well as the peripheral nervous system. This is going to be chapter 8. So we've got the nervous system and the brain. So, CNS, central nervous system. This includes your brain and spinal cord. Neurons and axons are bundled into tracts. Peripheral nervous system. Nerves, everything that isn't the central nervous system. Remember, central is brain and spinal cord. PNS, everything else. Neurons, axons are bundled into nerves. Nervous tissue, the body's primary communication and control system. Nervous tissue interprets and controls all sensations and movements. Functions are sensory input. It monitors stimuli occurring inside and outside the body using receptors. Integration, the interpretation of sensory input. So first it gets the input, and then it has to interpret it. And then is motor output. It has to respond. So it's a response to stimuli by activating effector organs. So let's look at the cytology of nervous tissue. This is going to be real interesting. So there are two types of cells within the nervous system. We've got neurons and we've got glial cells. Neurons are electrically excitable cells that initiate, transmit, and receive nerve impulses. Glial cells support and protect the nervous system by providing structural support, forming myelin, modulating neurotransmission, and playing a role in immune responses and tissue repair. So yeah, that's myelination, nutritional support, blood-brain barrier formation, BBB, and then structural support and protection. Neurons, they're electrically excitable, initiate, transmit, and receive nerve impulses. They conduct nerve impulses from one part of the body to the other. They feature high metabolic rate, extreme longevity, and they are non-mitotic, which means that they're not relating or involved in mitosis. Neuron structure. So there are three main structural regions to a neuron. You've got the cell body, the dendrites, and the axon. Okay, here in this picture, you can see the cell body, which is going to be on the left surrounding the nucleus. It's going to be the big old pink thing. And then branching off the cell body, you have your dendrites. And then the main branch of that, that's where your axon is. That's where it's going to send information, send and receive info. And that leads you to the axon terminal. A good way to remember that. Terminal, that's the end. And yeah, so that's the neuron anatomy that we have. Now, the dendrites, those are receptors. They have a sensory input. And then the axon, that contains cytoskeletal fibers. No axon hillock. It transmits nerve impulses away from the body to other cells and has three associated structures. Axon collateral, which are side branches of the main axon. Axon terminals, which are fine terminal extensions at the end. And synaptic knobs, which are expanded regions at the tip of axon terminals. So synapses, those are specialized junctions of communication between one axon and another neuron, muscle cell, or gland cell. Synapses transmit signals, and there are such things as presynaptic neurons, postsynaptic neurons, and synaptic clefts. Nerves and tracts. Nerves are cable-like bundles of parallel axons in the peripheral nervous system. Tracts are bundles of axons in the central nervous system. Glial cells, aka neuralgia. Their function is to provide support, protection, and nourishment to neurons, maintaining homeostasis and facilitating proper nervous system function. There are 6 types: astrocytes, ependymal cells, microglia, satellite cells, and Schwann cells. So of the 6 types of neurocells, both the ones that start with an S, satellite and Schwann, those are a part of the peripheral nervous system. Astrocytes are the most abundant glial cells. A for astrocytes, they're everywhere. Those ones are versatile and they have many functions. They're highly branched and look like stars. And they have 4 main functions, which are to help your blood-brain barrier, strengthen and organize tissues in the central nervous system. They replace damaged neurons. They guide migration of young neurons. And yeah. Next is epidermal cells. They're ciliated cuboidal epithelial cells. These line the ventricles and central canal. They help create cerebral spinal fluid, and the cilia help circulate that fluid. It forms the choroid plexus, and the cerebral spinal fluid fills space in the nervous tissue and supplies nutrients to neurons. Microglial cells, they're small ovoid cells with thorny processes that touch and monitor neurons. They wander through the central nervous system and can exhibit phagocytosis, removing cellular debris in dying cells. So microglial cells, they're the cleanup crew. They're coming around, they're picking up the trash, and doing a great job. There are the resident macrophage. Next, let's look at the oligodendrocytes. They're large and bulbous branched cells associated with the central nervous system axons only. They processes and wraps CNS axons, forming myelin sheaths. Oligodendrocytes myelinate the axons in the CNS. Basically, concisely, that. One of these cells can myelinate many axons. Next, satellite cells in the peripheral nervous system. These are flattened cells that are around neuronal bodies in the ganglia, and they surround neuron cell bodies in the peripheral nervous system. They function similar to the astrocytes of the central nervous system. They keep neuron cell bodies separate from the bloodstreams. They separate and regulate nutrients. They're pretty important because neurons can't repair themselves if they get damaged, so they're very good at protecting. The neurolemmacytes, which are also in the peripheral nervous system, aka Schwann cells, they're associated with the PNS only. One or more of these cells per axon, and the function of the neurolemmacytes is to produce the myelin sheath, which is a good insulator for electrical activity, and they have similar functions as oligodendrocytes. They're vital to regeneration of damaged peripheral nerve fibers. And yeah, that's pretty much all that we've gone over for the nervous system so far, and that's all that we need to know for the second exam regarding that. And yeah, so that's the integumentary, the skeletal, the muscular, and a little bit of the nervous system for exam 2 in anatomy and physiology. Yeah, all right. Good luck studying, listening to this on your drive, and I'm going to go drink some tea because that was a whole lot of talking. Bye.